Anyway, welcome everyone, all you wonderful people who signed up for our 9th webinar. And today I have invited Dr. Windi Muziasari, who is a very, very special person because her knowledge is very, very particular in regards to antibiotic resistance, which is a huge problem in the world. And of course she's going to tell even more about it than I can just say here. But before we move on to that, I'll just tell you that of course we are recording the webinar and we will share it with everyone as soon as we can. And I'm Anne-Catherine. I'm the co-founder of Nordic Laboratories and DNA Life, where we support practitioners to get easy access to laboratory tests and food supplements, and we ship and drop ship around the globe. And yeah, we just try and make it easy to work with functional medicine or personalized medicine using both stool tests, DNA and hormone tests, even in one order. You can order from different laboratories. So next webinar is not going to be on Thursday, because on Thursday my son is graduating with his bachelor thesis. So I'm going to push it another week, and then so in 2 weeks time, not same place for me, but maybe for you, but at least on the computer, Graham Jones will entertain us. He is an amazing practitioner. He's in Stockholm, and he's going to talk about energy and mitochondria. He's going to talk about cell danger response and hypometabolism and so on, and really move it into a clinical aspect so that a lot of you get ideas how to help your patients even better, and with new ideas and new inspiration and so on around the mitochondria. Big topic, but a big topic. But of course we will send out invites as soon as we can. So then moving on to Windi, of course, who is just the sweetest person. And I met Windi about a year and a half ago. She visited me in Copenhagen, but already then she had met Chris, my partner in Nordic Labs and partner in life. And they had started working together with Windi's company, Resistomab, where Windi is the CEO. So, and yeah, so not apart from being this really, really nice person, she of course has this huge competence within her field. And I was just really honored to meet her and to get to know her and be able to work closer with Windi and understand her ideas. And I think actually Windi is like one of the top 5 researchers within her field in the world, which is pretty amazing, from this woman who has traveled from Indonesia and now lives in Helsinki. And yeah, so anyway, Windi, I'm really honored that you would like to spend some time with us here. Yeah, and yeah, you are to take the stage now so you can share your slides. And yeah, we're excited and we are listening in.
Yes, thank you, Anne-Catherine. And it is my honor also to be invited here. Thank you. I'm very happy to be here with all of you, and maybe we'll talk more about the antibiotic resistance and how I passionate to work on this issue. Yeah, okay. So yeah, my name is Windi Muziasari. Well, thank you for introduction before. I'm indeed originally from Indonesia, Bandung. If any of you have been in Indonesia, so still in Jawa Island. But now I've been living in Finland since 2010, or like 10 years ago, for my PhD at the University of Helsinki, working on antibiotic resistance since then. And I did my postdoc also in Indonesia. I mean, the project is in Indonesia, so that's why I realized that antibiotic resistance indeed is a global problem, especially in developing country where people are literally die due to this antibiotic resistance, you know, because most of the bacteria cannot be treated anymore. So I mean, the disease cannot be cured and people then die. So it's really bad situation. And that's one of the reasons why I built the company Resistomab to help like everyone to get the access to laboratory for antibiotic resistant monitoring. And that's why I get to know Nordic group also. And that's why I'm here. And yeah, so today I'm going to talk about the profiling antibiotic resistant genes in stool samples. And yeah, as I mentioned before, antibiotic resistant, it is a global problem. So some of you may know heard about the superbugs. So these guys, the superbugs, are bacteria that cannot be killed by antibiotics anymore. So this is really bad, right? Because a simply bacterial infection could kill us again, like diarrhea or simply wound or like, can you imagine like the life before antibiotics, like 100 years ago? That's really bad. The mortality of women delivered a baby can be 50% higher than 50%, no more surgery. So many other activities can be limited when we don't have antibiotics. And unfortunately, for the past 30 years, we don't have new antibiotics anymore in the, like developed. So it means that we need to be careful with the current antibiotics that we have, and be careful to reduce, let's say, to prevent the spread of this resistant bacteria. As we all know, for example, current situation, this COVID-19, this pandemic. So, like all of us forced to work from home and limit our relationship. So can you imagine if the pandemic of these superbugs with all bacteria become resistant globally? It means that not only working from home and everything, so really need to be, like all the disease cannot be cured anymore. So that's not like world that we want, I believe, learning from what we have situation now. So that's why this antibiotic resistant problem, of course, is a global issue. WHO stated as a global problem actually since 2015. And you can see now also more in news. Like for example, in UK, it's just mentioned that how this superbug spreading in hospital, not only UK but also whole Europe, and of course in US also, and not to mention in developing countries. And it just last September, there is an outbreak of these superbugs in Tuscany in summer, in many hospitals, in 18 hospitals, where 30 people died due to the diarrhea. And this diarrhea is simply caused by E. coli. I think, if you like when you travel, you get always this travel diarrhea. And unfortunately, this one caused by this superbug E. coli, where carbapenems antibiotics, or like the last resource of antibiotics to treat diarrhea, didn't work anymore, because the E. coli carry resistant genes and become resistant to antibiotics. And we hope that this not happen to every hospital. So what should we do then? Let's get to know more. That's why we need to get to know more this antibiotic resistance. So why bacteria become resistant to antibiotics? There are several mechanisms in bacteria. For example, first is efflux pumps. So bacteria has the ability to pump out the antibiotics from inside the bacteria cell to out of cell. And then bacteria also can produce enzyme that can degrade antibiotics, or can alter the antibiotics that is not then become deactive. And another method that bacteria has certain protein in their wall that didn't allow antibiotics enter the cell. So all of those three mechanisms and other mechanisms, everything are encoded with these resistant genes. So when bacteria carry these genes, means the bacteria can have the ability or mechanism to become resistant. The problem here that the resistant genes in bacteria can transfer between bacteria. So for example, again, this E. coli. So when in your body, for example, you have only a normal E. coli that, you can use normal antibiotic ampicillin or beta-lactam and they die. But then you get infected with this E. coli that's resistant to antibiotics. And this ability can be transferred to other E. coli. So it means all your bacteria or your E. coli in your body then become resistant. And then this gene transfer also can be happening in different bacterial species, means from E. coli to other bacteria like Clostridium or Enterobacteriaceae. So means that the bacteria, not only the same species with different kind of species, like in our gut, in the food, in different kind of environment, then can spread this resistant genes. And that's why antibiotic resistance become widely spread and globally. And there are different also mechanisms like how the bacteria can be transferred. I mean, how these genes between bacteria can transfer. The first one is called transformation. So when resistant bacteria died, the genes release and can be absorbed by other bacteria. Another method is called transduction, where bacteriophage are viruses that attack only bacteria. So when bacteria infected by this phage, they carry the resistant genes from this infected bacteria, and then the phage release infected another bacteria, carry the genes. So that's why new bacteria will become resistant. And then another one called conjugation is actually bacteria mating each other and they are transferring the ability of resistance to another one. So this one actually the most common how the resistant genes are transferred between bacteria. And if you know that all these genes can be transferred between bacteria in different way, so this how the resistant genes then spread in whole system, especially in the environment. So when we use antibiotics, either as a medicine for us, resistance will develop in our gut and then release to the wastewater treatment. The use of antibiotics is not only in human. Actually, antibiotics produced in Europe, more than 60% are used in animals. For example, in aquaculture, for our pets, and also in animal farms. So those also, when antibiotics are used in animals, the resistant bacteria develop in their gut, and of course are released to the waste. And then if you use the waste as a manure in agriculture, it will transfer to the soil. Same in aquaculture, it transfers to our water environment, and then from human activities, from wastewater treatment plant, we release also to our water and soil environment. And then again, when these genes are spread in the environment, as I mentioned, the genes can be transferred to the bacteria in the environment. In that situation, they're not maybe dangerous because bacteria in the environment not necessarily kill us, but sooner or later, the more we interact with the nature, right? I mean, we cannot avoid to be like swimming or like when we, like play with the soil, or like it's just part of our life in the daily, or like through food, when we use, coming from the food when they've been treated so much with antibiotics. So this kind of thing, these interaction are happening very, like in the daily life, and the genes are transferring between bacteria from different kind of environment, which is eventually will end up to our bacteria, in our bacteria. And what we are worried when these genes are transferred to pathogens, when pathogens are cause disease to human and also to animals, then it's not good. So what we should do, we need to prevent the spread of this resistance, not only like in the clinical setting in the hospital, but also in the bigger setting, like in the daily life, in our life, in the normal, in our daily life. And so yeah, now I will get more, explain more the detail, like how we will do to profile the resistant genes, especially in the stool samples. So the problem now, as I mentioned, that these resistant genes are like carried by every type of bacteria. And when we are talking about bacteria, most of bacteria, actually more than 99%, cannot be grown in the isolate bacteria, like when we are usually normally isolate the pathogens in the clinical. So bacteria in the environment, or bacteria in our normal flora, normally they cannot grow in the laboratory plate. So what we should do is we need to do the molecular method, where we collect all the DNA. So we do total DNA extraction from any samples. In this case, let's say our stool sample. And then I'm using the SmartChip QPR system. So some of you may be familiar with the qPCR. So it's a method that you can detect and quantify the presence of the genes in the sample. The difference of the smart chip, you can analyze up to 384 different genes in each sample. So it means that we can do high throughput. You can imagine like 384 genes, because a standard technology usually few genes, like maybe up to 10 genes, 6 to 10 genes in one analysis. But using the smart chip, we can monitor up to 384. So you mean that very comprehensive and much more faster, of course. Since the data that we produce also pretty comprehensive, so we automate the analysis that we can produce the data more, hopefully easier to understand. And yeah, hopefully this is easier to understand. It's pretty comprehensive because it's 384 genes. So 384 genes that are encode or like giving resistance to bacteria, to different kind of antibiotics that are used in human. I will maybe short them to detected only. So it's like much less. You can see here that the gene's name in the horizontal line, and the classification of the genes to different type of antibiotics. There's tetracycline, MLSB is macrolide, beta-lactam, sulfonamide, aminoglycoside, you know, all the type of antibiotics that are currently used in human and animals. And the left side here are the stool samples from different person, and different line means different sample, stool sample, or collected in different time. And you can see also here the heat map color. Red, of course, means like bad. There means like abundance. And then blue means low abundance, but still means the genes are present and detected. And you can see also some paint gray color here. They are still detected, but below detection limit. So means that they are present, but in very low abundance. And white color means they're not even detected at all. So there's no positive signal in the system. As you can see here, that the profiles of, it seems like if you just see the red color, the profile of stool samples are pretty much different. The first one you can see here, I put it in the first, are patients that are diarrhea patient, like quite heavy diarrhea, that are having antibiotic treatment. So the first one. And then the second and the third are, we call this a super pool because they are used for the donor, for the fecal microbiota transplant. Some of you may be familiar with this treatment. So you know that require donors that pass through so many different kind of tests to be considered healthy and like become super pool. And you can see here also they are having they are also resistant genes here. And another sample here, like Helsinki, is actually a different person who's living in Helsinki, and then Copenhagen, living in Copenhagen, living in Reykjavik. And also I add a dog by actually a person in sample number 6 here, and then urban rat, like in Helsinki area. So you can see here, like it seems like the most abundant one, which is tetracycline resistant. It's interesting to see here tetracycline, because we're not using tetracycline anymore, never and ever. I think especially in the Nordic countries, tetracycline has never been used more than like 20 years ago or more than that. Tetracycline I only still use in animals. So if we, of course, like I don't have, like there is no such a real research to prove that these resistant genes are directly from animals or like from the food that we eat. But you can see here that how spread of the resistant tetracycline genes in our gut when we don't even use tetracycline. And then another, so you can see, now I can maybe show a bit more detail about the genes. Beta-lactam because that's the most common antibiotics that are used in not only in Europe, but globally. So it's very important antibiotics. And this is the story that I mentioned also in the beginning, where the diarrhea happened. So we are in a good situation here, because the genes that found in the Tuscany case, where there's outbreak that caused 30 people died, is called BLA and MDM1. It's called actually New Delhi resistant genes, because it's first found in New Delhi, and this NMDM1. And actually, when bacteria carried BLA and MDM1 genes, they cannot be cured anymore, unfortunately. So that's how important we need to do this kind of profiling the genes, because when we just see only they are resistant, but we don't see it into the molecular level, we don't know like how we can cure this, the resistance. Now we can see here that the most common beta-lactam that found seems like in our samples now is only CFXA. Indeed, the CFXA are the most genes that commonly found in the clinical setting. And you can see here the color is also like quite like, it seems like all bacteria are resistant. You can see also almost all of us carry, even dogs also carry CFXA, but then less abundance compared to the patient when they are really having the antibiotic treatment. So this is kind of, in my opinion, this is very important when we have this carry these genes. So when we are healthy, maybe we don't have any problem. But then if, of course, we don't want to have that happen, but if we got, let's say, Clostridium CDI infection, for example, the infection, that maybe not a good to have these genes present in our system. So, but unfortunately, it seems like these genes already spread everywhere. So that's why the important to prevent the spread of these genes, like not to be carried by all of us anymore, because if all of us carry these genes and spreading to more people, so the antibiotics to certain type of antibiotics will not effective anymore. The same as I mentioned, this BLA and MDM1. If every of us carry an MDM1, means that carbapenem's antibiotics cannot be used anymore, or will not be effective anymore. So hopefully this situation won't happen, and I don't know, we can to prevent the spread of this. And now we can see here that only CFXA gene that are most common found, but the rest are still very, less abundance, and not everyone carry the genes. And then another one, if we got more detail on vancomycin resistance. So vancomycin is very important because it's the last resort of antibiotics that we are using. So it's like the third generation. Actually, in Indonesia, the use of vancomycin is very, very rare because it's expensive and it's the last resort. So that's the only antibiotic that very strictly used in Indonesia. But of course, like in Nordic countries in general, vancomycin use are quite often, and especially used to treat this diarrhea patient. As you can see here, that they have more vancomycin compared to the rest. Of course, most of us also carry different type of vancomycin resistance, because in general, our microbe, like natural flora in our gut, they are carry vancomycin resistance. It's natural happening. So we don't need to use even vancomycin, but this resistance will be there just because natural flora in our gut, they are resistant to vancomycin. But you can see here, when the patient using antibiotic like vancomycin, more resistant genes found and a slightly more higher abundance. So let's to wrap up, like kind of more like summarize. So the number of the genes detected in individual human is usually between 30 to 70 genes. And from number of detected genes, you cannot really see the difference between patient, the superpool, like let's say, super healthy and the healthy, general healthy people. Maybe it's almost the same with the rat also, if we just focus on the number of detected genes. But this is very important to see. That's why I present previously, to see like more profiling, like the profile of the resistance. And it seems like beta-lactam indeed are the most common genes found in our gut system. This is, I would like to more emphasize some of the more detail in the individual stool samples. So sample number 4, we have like 5 different stool samples. It was collected in different time during for the past 2 years. So I saw here like interruption, or like in the red arrow here. These interruptions means that the person was traveling. So the sample 4B were collected when the person just returned from the traveling, and then 4C also when the person returned from another traveling. So it seems like after you travel, the genes seems like increasing the number of resistant genes. But then the sample for D and for E, the person was not traveling at all. So then the seems like the number quite constant. And then, but the difference here in the sample number 5, there is this quite another disruption, but this is not because of traveling, but the person was sick and had antibiotic treatment, like oral antibiotic treatment. So you can see here that the number of the resistant genes does drastically decrease. It's interesting, right? Because use antibiotics, why the number of resistant genes not increasing? Instead, this one is heavily decreasing. So this because when we take antibiotics, we kill most of the bacteria in our body. So as I mentioned before, there are certain bacteria that are naturally carry resistant genes. They might die. So then means the level, let's say the profile of the resistant genes decrease. But you can see here in the abundance that the same when the number of genes decrease, but actually the level of resistance are increasing, because the use of antibiotics means that the more genes are, there should be more resistant genes in terms of the abundance, but not the diversity of the resistant genes. So this way, it's also explaining in the beginning why between patient, if you just see the number of detected genes between patients and healthy people, there is no difference. But then you can see the difference actually in the level of resistance, the abundance. Level of the abundance are much higher when you are taking antibiotics compared to the healthy one. And sample number 8, this is, I just want to show that it's coming from the same sample, but using different method. Like the first one, when you collect the sample fresh, you know, like fresh poo and freeze them directly and goes for analysis. So this is, and then where the sample B and C, when they're collected using the normal stool sample collection, like when you have a stool test. So when there's the liquid protection. So it seems like using those methods, not really significant change. Of course, a few genes detected, few genes less detected, but it's not significantly different. So it means that different method for collecting the stool samples not affect to the profile of the resistant genes in our stool. This is more maybe concern for the technical people. And yeah, so but now we know that our stool sample carry the genes. So can you imagine if you're in the hospital now, all the stools are released to the wastewater, and means that this, the wastewater in the hospital are you know, all the resistant reservoir will be there. And that's why important to know the level of resistance in the hospital, and especially for the infectious disease control unit, to make sure that the resistant genes not spreading. So of course we need to monitor also to all antibiotics that are used in hospital. So what we do actually in Resistomap, we collect the wastewater sample And we analyze, we provide the result like less than a week. And this is the result in the wastewater. So there's 2 hospitals, Hospital 1 and Hospital 2. Hospital 1 is actually big hospital, more than 500 beds. And Hospital 2 is slightly, not slightly, like small hospital, like around 120 beds. So it means like small, but you can see the profile here. More genes were found in hospital, and you can see actually small hospital has more abundance. Like you see more red colors and more genes found, especially in beta-lactam and vancomycin also. So now I focus more in the vancomycin and beta-lactam. As you can see here, Hospital 2 has more resistant to different kind of vancomycin, which is meant like almost different type of vancomycin that we analyze present. We have around, I think, more than 40 different kind of assay to detect more than 40 different kind of resistant genes to vancomycin. And out of 40 assays, I think this is around 20 or 30, 20 genes are present in the hospital. So this is like why hospital wastewater are very important to be monitored to understand how the level of resistance in the hospital. And then as we can see here, big hospital not necessarily, even though the use of antibiotic the same level, but it's not necessarily has more resistance. It seems like the level of resistance more associated with the number of bacterial infection case. And the same case with the beta-lactam. You can see here like more variation. For beta-lactam, we have more than 90 different kind of assay to detect more than 90 different kind of resistant genes to beta-lactam. And in this case, you can see here like how many genes are present and pretty abundant. And so this is very important. And you can see here also, there are very different type of CFXA, of course, here is the most abundant, but you can see there are more different kind of beta-lactam resistant compared to the healthy human. Of course, you don't remember what the resistant genes we have before, but if you go back later on, I can show that the profile in clinical, they're pretty different. So this is why in the hospital, that we need this type of this molecular resistant genes that we need to prevent spreading to our environment and prevent this enter to our body. And so yeah, to summarize, in the wastewater, we found actually more than 100 genes, and even can more than 200 different kind of resistant genes. So hospital wastewater can be the reservoir of these resistant genes. And that's why it's very important to treat this wastewater before release to the environment. And unfortunately, in most developing countries, many hospital waste, they're not treated. They just release directly to the river. That's really not good. So this is why it's very important for us globally to help each other, because if it happens somewhere, unfortunately, our water system are connected in the sea, or we travel, or even let's say we prevent the traveling, like due to this COVID-19, like less people are traveling, but still birds are migrating, still fish are swimming in the ocean. So those wildlife also can help transferring or like spreading these resistant genes. So that's why it's very important, like we prevent this kind of spreading of resistant genes from this type of reservoir, like hospital, like animal farms, before this entering to our environment. And again, the most common genes found is the beta-lactam, but then vancomycin. So this is not good in this case. So yeah, now a little bit more resistomap then. Why then I came up with this resistomap? Because as you can see there, to provide this type of result is require state-of-the-art technology in molecular laboratory lab, which I know that not every hospital or every research or every lab can have it. And that's why the idea of resistomap may be similar value with the Nordic groups, like to give this access to everyone to get that this access to this technology. So you don't need to build your own lab and no need to be expert in antibiotic resistance, because we provide all the analysis and all the service laboratory, and we provide also fast result that you can, when you get better data, so you can have a better risk management. And I myself, as the founder of Resistomap, so one of the reason why I'm so passionate about this issue, because I've been working on this for more than 10 years, since I even started my PhD. So, and I've been working on this and focusing on profiling antibiotic resistance in the environment. And that's why, of course, I start to become more expert globally, because not so many people maybe just focus on one thing. And while I've been working on this, especially smart chip qPCR technology, like to have this comprehensive analysis of resistant genes since like 2014, when the first technology was developed. And that's why I want to bring this know-how technology to be available for everyone, or to be used. Of course, that's the idea of any research, right? Or any kind of technology. And together with my co-founder, William, who are more computer guy, like to automate all the analysis, to make all the process faster. So it's so nice like to have him on board to make all the process faster. So you can get all those results. That's why all this comprehensive monitoring up to 384 different genes, like in only one day. And of course, we have more team here, like working in the laboratory, and of course in the management also. And then here are our advisory board. We have Chris, of course, from the Nordic groups. And then we have also Veli Jukka from the hospital, Helsinki Hospital. He's also the head of the infectious disease control there. So that's give us more information, the important, of this resistance. And then we have Professor TJ, who has more than 40 years working on antibiotic resistance, especially in the environment. He's like the father of antibiotic resistance in the environment for most of us. And he's actually the first, this technology, the use of smart chip technology to profile antibiotic resistance was developed in his lab. And I was spending also visit research in his lab to understand better about this technology, and therefore I can build this Resistomap. Having this information, so this knowledge is not just came out recently, but it's accumulation of more than 40 years of research, and unfortunately, indeed, it still stay mostly in research. And that's why I want to bring this research to be available and to be used by every practitioner or everyone, even you. Like you can just use this information and data to help some problem in your case, because having more better information, more comprehensive information, hopefully can help better for the how to make decision and how to build some, for example, some policy or some kind of potential, or to evaluate some treatment. And yeah, Resistomap not only working on the stool samples or wastewater, but also different kind of samples. So we profile resistant genes because we want to, our vision is like a global monitoring of antibiotic resistance, including in manure, of course, when this animal, in irrigation water, in, of course, in this aquaculture, chicken farms, and even in the seawater, in the beach where when you swim, and even in the zoo, we have orangutan, and then of course sewage system, even to the wildlife, like we have from the Galapagos Island. So you will, it's like how amazing, I mean, it's not amazing, but how bad our humans are, because Galapagos Island, those remote area, and we study the seals who live in Galapagos, and unfortunately they carry resistant genes that are clinically to human. Like how on earth seals could have resistant genes, right? Because they don't use antibiotics. They've never been in the hospital, but they carry resistant genes also, because humans traveling there, and of course the pollution that we produce. So these resistant genes are spreading already pretty much everywhere, even in the remote area. So that's why our patients in Resistomap is like to have a robust monitoring, like this comprehensive monitoring, so we can identify and characterize the source of resistant genes, and then when we know where's the source and we identify where is the potential source of this antibiotic resistance, hopefully we can do some intervention there or some mitigation in like more efficient way. And hopefully, of course, our main goals is like to make antibiotics more efficient a bit longer, until hopefully we can get new antibiotics in the future. And that's why as a take-home message, please use wisely antibiotics. It's very important. Each of us individually aware the impact of using antibiotics, and especially in animal farms. So how important it is that you care about the organics, and of course the use like to take care of your microbiome system, like to be nature as possible. And then the important in this case, like to monitor not only in clinical setting, but also in the more One Health system, where involve human, animals, and our surrounding environment. And yes, thank you.
Thank you, Windi. So now what happened in Copenhagen last weekend was that the journalists got hold of finding out that they were going to take wastewater from Copenhagen and put it straight into the ocean 4 kilometers away from Copenhagen, in between Sweden and Copenhagen. And that was leaked out Thursday, that this had been approved by the environmental and technical department who had approved that this was going to be okay. So I didn't hear about this until Saturday in the news. And I was telling my husband, like, we need to, this is going on, it can't happen. All these antibiotic resistant genes, and Chris, my husband, got on to Windi and like, Windi, what should we do? And Windi then pulled some strings and got hold of some professors in Denmark and what have you. And then they were gonna open it up Sunday morning, but then they said they were gonna delay it 24 hours. And now they're just saying they delayed it until the autumn. So I hope that Windi, together with her team of experts, are gonna like try and prevent this, because basically it will be stool coming out with antibiotic resistance into the ocean, and what impact will that have on the wildlife in the ocean and so on. So that was what happened last weekend, and it just matched, and I just wanted to tell this story that Windi is one of the go-to people when it comes to this issue. Now, Windi, I know a lot of people listening here are practitioners, and they're gonna ask, so how can we prevent ourselves and our patients from getting all these antibiotic genes into us, or getting infected with these antibiotic genes? Have you got I know you're careful, because there has to be a publication and so on before you say too much and so on, but come on, share a little bit with what you're thinking and what your gut feeling is on this, please.
Yes, yeah, indeed. We need to be careful in this case, because it's still in the research. So we most of the researchers are still trying to figure out how like we end up with these resistant genes, as you saw now. Like, even we are healthy, even the super poo, they have resistant genes quite high too. So as I mentioned also in the beginning, it's more likely than from our foods, because foods are coming from where we daily basis got interaction with outside. food is, for example, in the first, and how important to protect our what you call this, like natural flora of our microbes. So healthy food, you know, like that's why it's nice that in Nordic groups, like you have like promoting the healthy life and have more nature and probiotics and all of this. So it's very important. I agree in this case, like to be careful of what you eat and understanding where the foods come from. So if you know that the foods are, for example, coming from, let's say, of course, not necessarily if you're vegetarian, you might think that, oh, I don't eat animal, so I should be okay. Not necessarily like that, because even the vegetables are used, you know, fertilized with the manure. And where's manure from? They're from animals, mostly, if you want to have the, you know, the most better fertilizer. So that's why, like, so this system is very important. So I think it's important for every individual to understand where the food that you are get from. I know from most of you who lives in Nordic countries, I think we kind of good in this case, because the food control are very, very strict, as far as I know, at least in Finland where I live. Also, it's very controlled. The use of how the use of antibiotics in animals is very, very strictly controlled and very limited. So it's a bit easier, at least for me, to find the food. But of course, if you live in a country where there's no control, try to be more, go to the nature, like more like small farms, or you know better the person, or you know where that's from, not like in the big kind of farming system where antibiotic use is like put together into feed. That's not the best option. And of course, also, like, there's a study now, this is already published, actually coming from mother when deliver the baby. So how we get also this resistance is already there since we were baby coming from your mom. So what we have now is maybe coming from our mothers, and you know, from the mother, from the grandmother, and kind of legacy. Maybe not from the grandmother, because at that time there's no antibiotics, but at least our grandma and our mom, that the legacy, they've been using so much antibiotics, and then, you know, then came to us. So, but then how important, and in this case, the breastfeeding, as if you able to do breastfeeding, that if you don't, then maybe just help the baby to give a better flora, the better diversity. As I saw in the beginning, the more you have profile, like diversity, usually the level of resistance is lower. So if you have lower resistance, it's okay to have, you know, like different diversity of resistance, but as long as they're in the low level of resistance. Because when they are in the high level resistance, means that most of antibiotics then don't work anymore. So at least that's, let's say, like, I would do for me, I would do for myself, knowing all of this situation. And again, always remind people that don't use antibiotics just like that, even though when the doctor suggested to prevent, but then you don't have a fever, for example, like, why would I need to use antibiotics? Some doctors still maybe want to make sure the patient, like, to be okay, like to prevent, like, for example, you get your tooth, you know, like remove your tooth, and you get, like, maybe giving antibiotic just to prevent that, so you don't get infected. But I think, in my opinion, don't use it until you really sick, you know. So you just use antibiotic when you really need it. And of course, like, try to inform your government, like, this is so nice, like, coming from you and Chris, like, so very directly contact the of course, like, if you have those access, it will be great to directly contact the authority, to be, you know, carefully make a policy to make antibiotics, like, more strict and this kind of thing. So that's maybe, sorry, then it's getting wider.
Now, I know I asked you this earlier, but it's one of these questions that just, what about organic food? Now, it was interesting with, even if you're vegan, so would organic food be better? And I know you have a comment on that one as well.
Yes, I mean, organic food is good. Is good, of course. Like, I try my best also to get the more organic food, but then again, need to be careful, like organic food, like certified organic food, because when it's certified organic food means that they are very concerned about the, like they know that when they use the manure as fertilizer, they knew that there is level of antibiotics very low, and they have this certification of the qualification of how to be certified as organic farm. I believe Denmark is one of the leading on this organic food, and they have very good regulation and level of certification, how, like what things need to be tested to be called as organic farm. But I know there are some organic food that are coming from farms just because they use natural things. They use manure as a fertilizer, but they didn't also control where's the manure from. If the manure from the animal farm that use a lot of antibiotics, then unfortunately does not help you than having those organic farm. So that's what I mentioned, is like the importance of understanding where the food's from. And yeah, of course, then in general, organic farm should be good, like, because giving more you interaction with the nature to give more diversity in your microbes. Maybe some of you have heard about the importance that you need to be more, interact with the nature, not just live to clean, otherwise you develop a lot of allergic, because our body system start to like protect to like didn't recognize, let's say, our system. So it's very important also to be in more in the nature to get, even though we get these resistant genes from the nature, they should be fine, because well, you can see later in my presentation the profile between the healthy people resistant genes profile and then the clinical, when this wastewater from the hospital, they are different in the molecular level. So it's okay in terms of the resistant genes coming from the nature, because they are a resistant genes part of the nature. This bacteria has been evolved since millions years ago. Maybe some of you know also, right? Antibiotics is coming from nature. It's coming from fungi. It's originally, like, for example, penicillin, and most of antibiotics are coming from in the nature. So it's okay, of course, to have the antibiotic resistance in our system, but not the clinical one. So that's why it's coming from the organic, talking about the organic food, just be careful about like how organic is it. But then again, depend on your country. Some country, like again in Nordics, I'm promoting so much the Nordic products here, but I live here. That's one of the reasons I live in Finland, or it's because I knew that all the food are, like when there's organic, they are really passed through the certain regulation. So that I can confident, let's say.
Thank you very much, Windi, for sharing that information as well. And I think the practitioners listening in here who's working with functional medicine, which is not your field at all, but what you can read between the lines here with what Windi says is that everything we can do to support the gut lining, support the immune system in the gut, being in nature, get exposed to healthy soil, and of course the birth with the mother, and like that will probably get some research in regards to cesareans and not cesareans and so on as well, I'm thinking. But yeah, walk the talk and in regards to functional medicine, and hopefully that will prevent a little bit of this, even though we're still exposed, especially if they put out wastewater directly into where we are swimming or and so on.
That's so good. Yeah, I was shocked too. Like, what?
Yeah, crazy. So, but of course we will inform a lot of all of you people about whatever the development is and so on. So there is a question that just came up from Magdalena. Do you have any advice for patients treated with long-term antibiotics, like with Lyme's or tuberculosis? Actually,
yes,
I both have tuberculosis and Lyme's disease. Good question.
Oh, wow, that's tough.
I'm interested in hearing this.
Yeah, tuberculosis, Lyme, of course, is a bad, but I'm more in the tuberculosis because my uncle's wife and his 2 daughters passed away because of tuberculosis. was a very, so for me, it's like one of the enemy. But yeah, unfortunately, indeed, this tuberculosis in certain country is already resistant. So most of the strain that cause tuberculosis are already the strain that resistant to all antibiotics that we have. So if you get tuberculosis from this certain strain that already resistant to all the type of antibiotics, then it's more like about, you just prolong the life, because it means that there's no cure. The same like this COVID-19. So literally you just help yourself to be healthy, like help your immune system to fight this, because no more medicine to cure. In the patient treated with the long-term antibiotics, I know that, to my knowledge, at least coming from the research also, there is no such solution, because none of us produce the new antibiotics yet, right? Because there is no yet there's not yet any innovation, either new antibiotics or new system that can replace antibiotics. So I believe this patient who've been treated this antibiotics for many years, the person will develop resistance, because the strain, the more you get antibiotics, they get more like resistant, and getting more resistant and increasing. So you need more like higher amount of antibiotics, or change to the stronger antibiotics, or combination of different antibiotics. I think doctors know better about this, but I know that certain antibiotics will not work anymore. So in this case, I think that you need to be more healthier and try to, to fight this bacterial infection with your body system. Like, again, back to your, like we discussed earlier, like help more your body to produce a better immune system, a healthier life. So because when antibiotics didn't work anymore to treat this tuberculosis, then you don't have any option, unfortunately. That's why actually tuberculosis now is in WHO priority list. So if a company who are now developing new antibiotics, like try to find a new antibacterial compound, tuberculosis is one of the, priority list. So there are 10 priority lists now by WHO. Lyme is maybe because it's important, but it's only happen in certain area, to my knowledge, the Lyme disease, not like tuberculosis where globally are happen. So that's why tuberculosis is more like spreading globally and become a global problem. But of course, individually, But as a person, individually.
Sorry, yes? I'm interrupting. I think maybe the question is more like, when you have been on long-term antibiotic, will you then develop even more antibiotic resistance? I think that's what it goes more on.
Okay, yeah, well, that's I think that's how it is. Of course, I cannot say like everyone, because I knew that individually people, if they have changed a certain healthier life, so maybe they can fight themselves, so the resistance go back to the low level. I mean, the bacteria strain like killed, like really died. And then when you get new one, it's just the normal same, but not the coming from the same bacteria just because you didn't kill properly and they become resistant. So those one that will develop more resistant. So it's very important then when you get the treatment of tuberculosis, take until all the bacteria died, let's say. Like take it to the proper, not just like, oh, I feel better now, I will not continue using these antibiotics. Please don't do that. Like, please follow the doctor's recommendation, where you need to take antibiotics this amount for this term, to make sure all bacteria that cause this tuberculosis died. So then hopefully then you don't get more disease. And then when you get more, again, new infection is coming from just the normal one, not because the one that been survive from the first treatment and become more resistant. So that's maybe, is it clear enough, I guess?
Yeah,
just to do the difference. Yes.
And then there were some asking about probiotics.
Oh yes.
Like what is that research on using probiotics and less infections with antibiotic resistant bacteria?
I think it's hard to say. actually some probiotics carry resistant genes, as I mentioned here, but it's more like natural resistant genes. So it's not so much affecting on the resistant genes. But then usually probiotics, to my knowledge, it helps our microflora. Let's say if we have very strong bacterial community, and then we have these pathogens who are resistant come in, I think he will not, or she or whatever, this bacteria will not survive in our community because we have very strong. So it means that these pathogens, when enter to our system, they cannot fight, to get the food or to grow, because we have very strong community. And I understood in this case, probiotic help us, our microflora, to get stronger army, let's say. So when the pathogens or even just resistant bacteria come in, they will not survive. I mean, yeah, this just came, but will not infect us because we are very strong. So that's in my opinion, because that's why most of us take a probiotic just to be healthier in general.
Okay. There was one last question. If you know any centers working on creating phage therapy, I know of one. I can't remember what they're called, but you can send me a direct message and I can put you in contact with them. It's not something we work with in Nordic, but are you?
Yes, phage therapy, to my knowledge, I knew that now recently work in human, but it's a bit tricky because it required a cocktails system, because it may work in aquaculture actually. Phage therapy are really used widely in aquaculture because it's aquatic environment, so phage survive better. But in our system, I knew there's few cases successful. Is because it require, because we don't want to kill all bacteria, we just want to kill the pathogens, right? So it means we need to have a proper phage that only attacks pathogen, not all bacteria. So then I understood in human trial is not easy because humans are so complex, and when we take it, our system is not suitable for the phage. So usually require some cocktails, bacterial phage, to make sure. So then it's not specific in this case. Maybe it's better only when you have when you don't have antibiotics anymore to treat, then it can be. But it's my personal opinion. Of course, some people who are expert on this, they know better. But to my knowledge, it's worked efficiently in aquaculture.
I can't comment on the effect of the phage therapy, but what we actually do with one of the labs that we are working with and a practitioner is that they order isolates for phage testing. So we send, so when they can culture specific bacteria, they isolate these bacteria and we ship them off to this research company that then produces phages for the specific patient. But send me a direct message. I know a little bit about it, but I'm just really just the messenger in that process. But I'm happy to share a little bit more information about it. So anyway, I think it's time to stop. So I'm going to give you, Windi, a big virtual hug, and I'm going to give everyone a virtual hug. And then I'll see you. Hopefully you're welcome as well, Windi, in 2 weeks' time. But everybody else, I'll see you hopefully in 2 weeks' time, when we move much more into the clinical picture in regards to mitochondrial function in health and disease and cell danger response and so on. So I'm really excited to that, and we'll send out the recordings from the day as usual, and we'll send out the invite with a link there as well. So thank you very much, everyone. Take care wherever you are in the world. Bye.
Thank you. Bye-bye.